Hello and welcome to this presentation. I will talk about artificial intelligence in chemistry, discovery processes and tools of the future. My name is Andreas Bender. I'm a reader for molecular informatics at the University of Cambridge, as well as the Director of Life Sciences with Novisan in Berlin. Any statements during this talk are in my capacity as an academic. This is the outline of my talk. Firstly, I will talk about hypes and predicting the future. Then we'll talk about processes and the involvement of AI, and I will illustrate that an old process with AI is still an old process, so we need to revise whole processes in order to make use of new tools. I will discuss problems along the way, and in particular, this will be the data we have available. And I'll ask the question, so what next? So where are we today? Right now, we're in the third wave of computers in drug discovery. There was one in the 80s, one in the 2000s, and one is today. And I think a time for realistic assessment has come. So what was characteristic for those waves? In the first wave in the 1980s, that was a very graphics-driven wave. Structured graphics came up, Silicon Graphics was founded, for example, the first docking programs were published. So there was very much a wave driven by graphics as well as modeling structural information. Then there was a second wave around 2000. This is when bioinformatics became popular, as well as cheminformatics to a lesser extent. That was the time of the stock market bubble, for example, as well. So that second wave was much more informatics driven than the first one, which was more graphics driven. And we have a third wave of using computers in drug discovery today, and these are some of the headlines we see. And the question is, of course, some of the headlines might seem similar, but is the situation similar? So what is actually similar? What is different? And I think the data is different, for example. We have much more data. Data is better annotated, for example, in ontologies. On the other hand, the headlines sound similar. Yeah, and sometimes it makes sense to look at the headlines that were published in earlier times and to make a step back and to see what actually developed, what actually really happened in the future. So I'm old enough to remember the 2000 biotech bubble that involved, for example, the publication of the Human Genome Project in 2000 as well. So what was published just a year later? For example, this was a review publication in 2001 by Thomas Rice, who claimed that the number of drug targets based on the Human Genome Project will increase by at least 1 order of magnitude, and target validation will become a high throughput process. There will be more drug targets, so 3,000 to 10,000 targets compared with, at the time, 483. But if we then look further, 16 years later, estimates of drug targets put the number currently at that time at only around 667. So not all expectations become true. And the question is, of course, right now we have AI and chemistry. So what is more likely to develop into tangible approaches in the future? And where will more problems be encountered? So what do discovery processes and tools in chemistry look like right now? Processes grow out of a variety of different factors. The first one is, of course, technology. So this is technology that enables us to do something. And that is a pull factor in this particular categorization. Then we also have, of course, cost, societal, and legal environment. And this is what I would describe as a push factor. Costs, for example, want to go downhill. We have societal changes, for example, what is accepted in a society. And we have a legal environment that forbids, for example, certain types of, let's say, animal studies. So this is a push factor that drives processes into a different area. And thirdly, we also have beliefs. For example, beliefs into big numbers, for example, high throughput screening, as well as analytical deconstruction. For example, that we can represent a particular disease in a biochemical assay, and we can use that to screen a large number of compounds. Plus, of course, processes are quite a lot about history as well. If you have a process in place today that developed given those factors as well as other factors in the past. So these are the different factors we have that describe which particular process comes into play today. So it's technology as well as the environment, as well as beliefs, and all of that together forms a particular process. But of course, complex processes are often difficult or even impossible to change. So what are reasons for that? Many of those processes take place in big companies that comprise a large number of staff, and they find it difficult to change processes mainly due to two types of reasons. The one is inherent to the system, and that means if you change one part of a complex system and the whole system breaks down, then it needs to readjust over a long time period. So it's difficult to only change a small part in a complex system. The other part is, of course, the human side. Humans are sometimes resistant to change, in particular, it involves some fears of becoming redundant in the future or the like. On the other hand, what we can see right now is that there's clearly disruption on the way, and those is disruption that I would call simulations or data first companies. So what is different about those companies? They can build processes from scratch. So there's no process that has been in place for years or even decades that needs to be adjusted. That makes it much easier to engineer process. And the second level, they can also attract staff that is on board with processes from day one. So that avoids this human factor of resistance to change. And this disruption, of course, advantages newly built environments. And that refers both to the company level, startup companies, for example, as well as on the country level. So this can often favor countries which didn't have a certain pharmaceutical or other industrial setup before. But of course, the question then is, how do you actually use artificial intelligence to change the processes? And it's important to actually re-engineer the process if you have new tools available. Because otherwise, what happens is the following. You have an old process, you combine it with a new tool, but then you have an old process that is supported by the new tool. But that does not take full advantage of all the power you have available. This is illustrated here on those images. What you need to do instead is, you have an old process, you combine it with a new tool, then you have a new re-engineered process that can take full advantage of the new capabilities. But that requires rethinking the whole process. What does that mean in concrete terms? I would like to give an example from drug discovery. Of course, there's not only one way how drug discovery projects can be run, but still there are some fragments, some pieces that occur in many cases. So they are illustrated here. So in many cases, you start with an anecdotal piece of knowledge as a starting point. So let's say a paper that describes a gene involved in a particular cancer. Secondly, you do a brute force high throughput screen to generate a molecular starting point, such as a hit. And finally, you optimize that compound by expert knowledge. But you see here that there's no decision making that really involves data at its basis. On the other hand, what you can do as an alternative, where you have sufficient data available, you can do the following. You can move to a data-driven starting point. Say we have a knowledge graph as a starting point of suggesting a particular compound for a particular indication. And then afterwards, you can perform an optimization based on data and, of course, expert knowledge. So I'm not saying that expert knowledge is irrelevant. However, even as an expert, it is difficult to compile all the information, all the data from a given project in one's head. So it's good to combine both the data side as well as expert knowledge. Of course, and that is important to keep in mind, there are always limitations of data, and there are two different types of limitations of data. The first one is limitations in quantity. This is actually, although it sounds very dangerous, the lesser of the two factors. That can be overcome by screening and generating more data points. However, what is a bigger problem in principle is the predictivity of a particular assay. We need to know what to measure in order to measure it at a sufficient quantity in the next step. And one of the reasons for that is the data is very different in the different fields we operate, even within chemistry. And this is what I would call data, the sometimes ugly child. What do I mean by that? I mean by that there's a core difference between different disciplines, and for example, between materials and drug discovery. In materials, we, at least in many cases, know what matters. Let's say it's a band gap of electronic structure, or it is a particular interaction between a catalyst and some substrates and so on. And in those cases, we can, in many cases, measure what we need to measure, we can determine those endpoints, and we can feed that into computational models. On the other hand, in more biologically oriented systems, such as drug discovery, we rarely know what matters, and most of our data comes from proxy assays. So what does that mean? For example, for a particular type of organ toxicity, we don't understand all the mechanisms, so we measure endpoints that are only vaguely correlated to the actual endpoint of, for example, liver toxicity that we're interested in, and then we're trying to piece together the particular pieces of the puzzle in order to get an idea of the endpoint of actual interest. And you see there are two different areas that behave quite differently, fundamentally differently even. Some are more physically oriented, we know what matters, we can measure it, we can build models because we can generate data in the first place. On the other hand, in other areas, more biologically interested, we often don't know what really matters. We have a wide variety of factors that influence the assay setup such as concentration, time point, the biological system, and so on. And in this case, most of our data comes from proxy assays that only resemble the endpoint of interest. And in those cases, it is much more difficult to generate data and to build models for a particular endpoint that really is crucial for decision making. That's a huge difference for AI in different domains when it comes to generating data as well as building models. I can recommend this article, Can a Biologist Fix the Radio? That really in vivid terms illustrates the differences between the different domains when it comes to both explicit representation of information as well as quantification of pathways versus electric circuits, for example. This particular behavior of data in different disciplines is illustrated here. At the top, we have the image domain, and we have in the different columns the object, the representation, the model, and the object label. And in particular, you see here that in the image domain, one of the areas where artificial intelligence really made great progress, we have a good representation of the object, in this case in pixel format, a model such as a convolutional network that can detect edges that are relevant for object classification, and we have an object label that is largely unconditionally assigned. And this is also the case in many areas in the more physically oriented chemical domain. However, in the biological domain, unfortunately, we need to deal with many problems, such as the conditional assay labels. For example, toxicity depends on dose, and so on. On the other hand, some aspects of representing chemistry are common to the domain, such as how to deal with conformations, tautomers, and so on, how to choose molecular descriptors, so these are common to a wide variety of different areas. Still, what we can see here is whether we can assign an object label in an unconditional way, a cat is present or not present in a picture, or in a conditional way, whether a compound is toxic depends on dose, really influences our ability to build machine learning models. One example for that is adverse drug reactions. So there are computer scientist approaches that ask, does a drug cause an adverse drug reaction? Yes or no? But of course, if we look into it in more detail, we see that's hugely simplified, because all of that depends on a genotype, the endotype, the dose, the dosing schedule, co-administration of other drugs, sex, duration of treatment, and so on. So in many areas, such as more biologically oriented chemistry, drug discovery, chemical biology, it's actually very difficult to label data. And so does a particular drug actually cause an adverse event? That's very difficult to annotate. It's very difficult to train models with the resulting impact on our ability to use AI in different domains. So our understanding of predictive endpoints influences our ability to generate predictive data, the right data for the right purpose, and that influences our ability to train predictive models that can be used for decision making. And this is something we discussed in a recent article on drug discovery today. Right now there's lots of attention on methods, computational methods, that might involve methods or combined methods and representations to end-to-end learning. On the other hand, all of that decision making is based on data, and actually even more fundamentally is based on a question or hypothesis. And the method cannot capture what is not in the representation, and the representation cannot capture what's not in the data, and irrelevant data does not answer the question at hand. So we need to think this pyramid from the bottom. We need to ask a question or hypothesis first, generate the right data for the right purpose, then worry about the representation, and only then, at the end of the pipeline, worry about the right machine learning method. And this is not done in many situations today, but we need to reconsider this pyramid, and that is based on a question and on data, even more than we do right now, in my opinion. So what next? We need suitable data to support our processes. To achieve that, we need to understand which data we need and which data we need to generate. There are differences between disciplines, for example, materials and drug discovery. In order to make use of AI in chemistry, in many cases that requires a re-engineering of processes in areas which we sufficiently understand. That starts with the generation of the right data and a problem that can be answered with the data available. So we need to have data for the right endpoint that we need for decision making. And this is in many cases, such as more biologically oriented disciplines, not really the case today. But of course, I don't want to conclude without referring to the bigger picture. AI is where it is also due to human psychology. For example, hype brings you money and fame, realism is boring, there's fear of missing out, all others do it, so we need to do it as well. And beliefs often drive decision. Maybe someone has the secret sauce, which is unlikely though. Everyone needs a winner, especially after huge investments in a particular area. There's selective reporting of successes that leads to everyone declaring victory, but in reality no one knows what's actually going on. And scientifically, it is difficult to really advance a field if there's little comparison of methods. So it's difficult to judge how good we actually are today and how much better we are today than we were yesterday. So to summarize, processes grow out of available tools and taking existing circumstances at a time into account. When tools change, processes need to change, and this has only very partially been achieved today. So we should not support existing processes with AI, but rather rethink processes from scratch. Areas hugely differ when it comes to data, the amount, the labeling, and the predictivity for decision making. In those areas with higher predictivity, better ability to label, and most data will benefit most and earliest from AI. Thank you for listening.